Hello friends, welcome to BMH Learning. In this video, I'm going to talk about enzyme multiplication immunoassay technique, commonly known as EMIT. First, let's talk about the principle of EMIT. In order to understand EMIT, we need to remind a little bit regarding the interaction between enzyme and its substrate. So, enzyme is a protein that has a catalytic site called active site. Now, the enzyme shows its activity when a specific chemical binds to this particular location present on the enzyme. The chemical that binds to this particular site is nothing but the substrate. So in this immunoassay, we use enzyme substrate system such that when the enzyme and substrate bind to each other, the enzyme brings about a change in color of the substrate. After this, the substrate is released. Now the active site is empty. So that means another substrate molecule can bind at this position. In a similar manner, again the enzyme causes the substrate to undergo a color change. And then the substrate molecule is released to clear the active site, so that next substrate molecule can interact with the active site and follows the same fate. The process of substrate binding and releasing will continue till the active site of the enzyme keeps on getting the substrate molecules. And a time would come when there would be so many substrate molecules in the solution which have already interacted with the active site and hence have undergone a color change. The change in color in these numerous substrate molecules cause the solution to turn its color itself. And that's what we are looking for, because we use this change in color property in EMIT. The principle of EMIT relies on the enzymes with the target antigen attached near the active site. Now, the target molecule can be a chemical such as a drug molecule or a poison. This simply means here we have our enzyme, and let's say we are testing for poison X. So, we will chemically attach the poison X antigen somewhere here, that is in the proximity or near the active site of the enzyme. What we are going to do next? We add antibodies against the antigen attached to the enzyme. So what's going to happen? The antibody will bind to the antigen, and that's what antibodies do. But here is something important to notice. Because we have attached the antigen near the active site of the enzyme, the antibody, when gets bound to the antigen, actually blocks the active site. The effect is such that the substrate cannot bind to the active site anymore. So, because it's not going to fit into that location, it would not change color. So that means when antibodies are blocking the active sites, there is no change in color. Now let's practically apply this principle. First, let's assume there is an antigen in the sample. So we have our sample, and the sample has the antigen we are looking for. Now remember one thing here. The antigen in the sample is actually identical to the antigen we will attach near the active site of the enzyme. So what do? We add the enzyme having antigen attached near to its active site. As earlier stated, the antigen on the enzyme and in the sample are both same. Next, we add the antibodies. Now there is the real magic. Because both the antigens are same, so the antibody has the choice to either bind to the antigen freely roaming in the solution, or it can bind to the one attached near the active site of the enzyme. So, few antibodies are bound to the antigen attached to the enzyme, and some are engaged with the free antigens. That means not all the active sites of the enzymes are blocked. So these are the free active sites. Now we add our substrate. This will search for, of course, the active site. Because there are some unblocked active sites, the substrate would bind there. What would happen? Color change. And of course, there would be many of the likes of these color changing substrates. Eventually, this would give us a color change in the entire solution. Here I would like to tell you one more thing. It's about the importance of the intensity of color change. More intense and quick is the color change, more concentration of antigen in the sample. Now let's know what would happen when there is no antigen in the sample. So we will add enzyme into the sample solution attached with antigen we are interested in. Then we will introduce antibodies into the solution. So the antibodies would do their stuff, that is, bind to the antigens they are looking for. We can see all the active sites in the enzymes have been blocked. So when we add the substrate, it's not able to locate the position where it can fit itself. So there is no color change. That's all. Thanks for watching.